So this video will be all about how we measure the number of bacteria that are growing in a culture. And first thing you should know is that these numbers are always going to be estimates. There's just no way that we can count the trillions of cells that are found growing in a culture as it incubates. And so all of these are about estimating, so doing the best possible mechanism for coming up with the number of bacteria that are in a culture. And when we express the number that we find, it's always expressed as the number of viable cells per milliliter of the culture medium. And by viable, we mean living and reproducing. So these are the cells that are going to keep on undergoing binary fission and producing new cells. Okay, so the first technique is called serial dilution. So if you have a sample of a known volume, then you can take a milliliter, so you mix up the culture, this known volume of culture that you want to know the number of cells that are in, and you mix it up really well so that the bacteria are completely brought into suspension, because frequently the bacteria will settle down to the bottom of the culture or will be found growing along the glass sides of a culture medium. And you mix that 1 milliliter sample then with 9 milliliters of water. That's a 1 to 10 dilution. And you get that all mixed up. There's a special machine. We've got one called a vortex machine. And so you would spin those around for a couple of seconds to get everything really mixed up. And then you would take a 1 ml sample out of that 10 ml volume that you just have made. So you add 1 ml into 9 mils. We have a total of 10 mils now. Mix that up. Take a 1 ml sample out of that and put it into the next tube that's all set up with exactly 9 milliliters of water. So you got to be super careful that you use exactly 9 milliliters each time. And there are special pipettes, they're called, that produce a very repeatable volume. So you keep doing these 1 to 10 dilutions. So if this is 1 to 10 diluted in the first one, oh, okay, I'm sorry, this is our original culture here. So if our first one, when we mix that up, gets a 1 to 10 dilution, then when you take 1 ml out of this one and put it into the next test tube, then you get a 1 to 100 dilution, and then you take a ml out and put it in 9 more mils of water, and then you have a 1 to 1000 dilution, and then you have a 1 to 10,000 dilution. And so you would keep doing that until you reach the point where you believe that you've got a countable number of bacteria, and you are tracking the dilution factor each time. And then with that last dilution, or frequently people will do it with like the last 3 dilutions to make sure that they've got a countable size of number of bacteria in this last step here. Then with that last dilution, you take all 10 mils and you either spread it out on a solid agar plate, so a plate that's already been poured, and this week we're going to practice pouring plates, so you're going to get some experience with that. Or you can mix that last 10 mils with melted agar, and you melt it and then you cool it so that the bacteria don't get burnt up. And then before it solidifies, you can mix in that 10 ml volume, and then you pour it out onto a plate. So if you do it this way, it's called a spread plate. If you do it this way, it's called a pour plate. And then you incubate the plate at the appropriate temperature for the appropriate amount of time. Usually it's a 24-hour incubation, and either at 25 degrees C, 30 degrees C, or 37 degrees C. Those are the 3 temperatures that are most commonly incubated at. And then you can count the number of colonies that grow on the plate. And what you're looking for is a plate that has a countable number of colonies, anywhere between 30 and 300. If you have fewer than 30 colonies, then it's not, then you've over diluted. If you have more than 300 colonies, then you probably can't count accurately. And there are tools that you can use to help make this counting go better, but still it's tough to count a whole plate that has more than 300 colonies. And then, so Each one of those colonies that you see growing on the agar plate represents one viable bacterium in that final dilution. What we think of as a colony forming unit. So we know it's viable because it made a colony. If it didn't make a colony, it would have been an inviable bacteria. And then all you have to do is multiply the number of colonies at that final dilution times the final dilution factor, whatever that was, and that tells you then the number of bacteria that are found in 10 mils of that sample. And then all you have to do is multiply by the known volume of the culture. And generally, people will incubate cultures in a known volume so that they're ready to go. And 10 mils is a reasonable one. Or larger, at liters, if you're trying to really grow out a large number of bacteria. Okay, so that's serial dilution, followed by a spread plate or a pour plate. Another way to measure bacterial growth is to do a direct microscopic count. And so you have to use a very carefully calibrated slide that has, you can make out in there that there's like a cross-hatched pattern in the slide, that metal portion of the slide there. And that cross-hatch area will hold a known amount of fluid. And when you look at that, so you can introduce a little bit so you put on these special cover plates, and then you introduce a little bit of fluid in here, and that will wick up into here a known volume of the fluid that is holding your bacterial. And then all you have to do is count the number of bacteria that you see. And so here's what you might view through the microscope, and you would count those. In this case, it's yeast cells, but I took this from a website about a group that's brewing beer, and so they're counting the yeast in the beer that they're brewing, making sure that their counts are proper. So you then count the number of cells that are in a certain number of those squares, and then enter that number into a calculation, and that will tell you the total number of cells that are found per milliliter. So you can do direct microscopic count using these Petroff-Hauser counting chambers. They're also called hemocytometers because they're used to count red blood cells as well. So if you needed to check and see what somebody's red blood cell count is, you could use these hemocytometers or Petroff-Hauser counting chambers. Okay, so you can directly count the bacteria in a small sample, and then by multiplying up, you would then calculate the total bacteria that are found per milliliter, and then you can then multiply that by the number of mils in your culture, and then that would tell you the total number of bacteria. Then sometimes, the bacterial load in something may be so low that it's hard to count using either a direct count or a pour plate or a spread plate. Because perhaps the organisms don't grow on agar. So if we got too small numbers of bacteria in the sample, or if the organisms can't be grown, then using the most probable, it says probably, it should be probable, so put an E there where it says probably, we use this most probable number measure of bacterial growth. So first, you would do a direct count to get a rough estimate of the bacterial count, and you would use that number to help establish an appropriate dilution series, just like we saw with that first technique that I showed you. And then you would take 5 samples each of a 10, 1, and these are the usual dilutions, but a 10 to 1 dilution, a 1 to 1 dilution, and a 0.1 to 1 dilution. So one where you actually have more bacterial stuff than bacterial medium than you have water in that final dilution, and you place these into a test tube of broth with a tube that captures gas as the bacteria as they metabolize. Outside. Excuse me, my daughter came in looking for the cat. And I had already thrown the cat outside, so that's what that was all about. Sorry. So you would then produce these appropriate dilutions, and if things go well, then what you look for is growth in your tube and production of gas in the little capturing tube that you put inside the test tube to grow it out. And you use then the growth to no growth results in order, and then that winds up being analyzed in a statistical table to tell you the most likely concentration of bacteria. And that statistical table is on page, let's see, it's in the book, and it's on page 154. And so they give you an example here where at 10 mils you had 5 positive tubes. At 1 mil you had 2 positive tubes, and at 0 mils, or I'm sorry, at 0.1 mil you had 0 positive tubes. Oh, you know, and I got that backwards as I was talking about what this meant. This is actually a very, very small sample. So you add 0.1 of that final dilution mils to the tube. You would add 1 mil of your final dilution to the tube, and you would add 10 mils of your final dilution to the tubes. So usually you do 5 test tubes. One has 10 mils of the final dilution, one has 1 mil, one has 0.1 mil. And then you look at how many of the tubes show growth. So again, I turn you to page 153 and that table there. So in the 10 mils dilution, amount of the dilution that was added, in that 10 mil sample, we saw that all 5 of them show growth. When only 1 mil of the final dilution is put into the tube, we had 2 of them showing growth. And when you added 0.1 mils, none of them show growth. And so you would take that 520 number and look onto the statistical table that you have, and you follow the 520 line over, and you see that the most probable number is 50 bacteria cells per 100 mils of broth, or of whatever the stuff you were sampling. Most probable number is used primarily for water quality sampling and air quality sampling. So if you're concerned about having bacteria in the air or in the water, then you may be getting such low counts that it's difficult to do a direct count or to do a grow out count. And so so using most probable number will help you to get a good idea of the estimate. And this is all based on statistical research, and you know, comes pretty close to accurate. Okay, another way, if you have low concentrations of bacteria in air or water, would be to filter a large volume of air or water through a paper filter, especially designed paper filter that has a very tiny pore size. So you would push this known quantity of water or air through the filter, or draw it through the filter, and it would capture bacteria on the filter, and then you slide that piece of filter paper onto an agar plate and incubate it. And then each colony that you see will be representative of one bacteria found in that known quantity of water or air that's been sampled. So again, it's all about estimating. So if you if this was a sample after filtering 300 cubic feet of air, for instance, then you would count the number of colonies there. So it looks, I'm going to estimate that it's like 35 colonies. And so you would then be able to say that, you know, in 300 cubic feet of air we have 35 colonies, so per cubic foot that's however many. Okay, so you get the idea. And then a last way that we can do these kinds of estimates is by using a machine called a spectrophotometer. Spectrophotometers will shine a beam of light through the sample and then collect on the other side the amount of light that hits on a receiver on the far side of that sample. And so doing this, then you can determine how much of the light winds up being absorbed by the stuff that's in the broth. So the more bacteria that are in the broth, then the more light that will be absorbed. And so if you know that a density of bacteria in a sample will absorb X amount, you can then produce a graph of a known, so we would see absorbance here, absorbance, and no, actually, I'm sorry, I got that backward. This is absorbance. This is the amount of light that gets absorbed. So down here it's 0 light is absorbed, up here is 100% of the light is absorbed. And then you have a curve that you can plot your number against. So you do this known curve, you figure out how many cells are going to absorb, let's say, half of the light. So you know that figure. And then when you shine light through your sample to test its turbidity, turbidity is like the measurement of how cloudy the broth is, how much light the broth is going to absorb, then you can plot your numbers. We saw an absorbance of this, and you can say, aha, that is this number of bacteria per mil. So you have to develop a set of standards so that you can develop this curve. But once you have the curve, you can test any number of samples using that known curve. So it's a very rapid way of quickly estimating the number of bacteria that are in a culture. I said this was the last. Now I really mean it. This next one is actually the last. Oh, no, it's not. It's the second to last. I've still got 2 more. Sorry. Yeah, psych. As you know, when bacteria are busy metabolizing, they're going to deplete nutrients from the growth medium, and they're going to produce waste materials and byproducts that accumulate in the medium. So if we have a way of measuring either the depletion of a known nutrient or the accumulation of a known waste material, frequently wastes are acidic, so a pH change will tell you something. Oxygen gets used up by aerobic bacteria, a bacteria that need oxygen to live. Nitrogen gets used up by nitrogen-using bacteria. So all of those could be used if we have a way to measure nitrogen or oxygen or pH, and all of those are measurable. Then the speed with which the media is either depleted or enriched, if it's a waste material, can be used to estimate the concentration of bacteria. So the reduction in methylene blue, for instance, is one example of this kind of metabolism. So as bacteria use up oxygen, I think it's oxygen, there's some material that they use up, then the methylene blue changes color from blue to more clear. And that tells you a little bit about the depletion of the nutrient and the number of bacteria that are growing. Okay, so finally, here we are with the last one, would be to use a dry weight measurement. So you take a known volume of growth medium with the bacteria, and you either filter it or you centrifuge it. So one way or the other, you collect all of the bacteria on a filter, or you can put it in a centrifuge. This is a microcentrifuge, and it uses these little microcentrifuge chambers. You put in a known volume of your growth medium. You can spin that puppy really, really fast for a couple of minutes usually, and that forces all the bacteria down to the bottom in this little pellet here. If it's a piece of filter paper, you could then dry the whole paper and the bacteria on the paper, and then and if it's this pelleted form, you would decant the liquid and dry down all the liquid out of it, and then you can scrape out that pellet, or you can weigh the whole microcentrifuge chamber, and the additional weight of the bacteria then tell you how much mass of bacteria there is. And the same with the filter paper. You just dry the filter paper with all the bacteria on it, and then weigh the filter paper before you put the bacteria on it and after you get the bacteria on it, and then you can estimate the number of bacteria based on the number of cells there are per microgram of the pellet. Okay, so lots of ways, but again, they are all estimates. They're not 100% accurate, but they will get you close enough to being able to estimate the number of bacteria that are found in your culture. You know what to do if you have questions.